Good afternoon. Welcome to RSSL first webinar of the Pharmaceutical Impurities webinar series, Nitrosamines Analysis, Better Understanding the Risk. Now, before we start, just some housekeeping. If you have any technical issues, please send a short message in the questions box on the right-hand side of your screen. The team will then respond to you directly and try to resolve your issue. Please be aware that we are working remotely, so sometimes there may be a slight delay. During the webinar, RSL would like to invite you to ask as many questions as possible, and also using the question box on the right-hand side. At the end of the webinar, time permitting, we will try to answer as many of these as possible. Any if we don't get around to, we will answer offline and share them with you all along with the slides from today's webinar. And at the end, please stay on the webinar. So, because we're running a little promotion, which I really like to tell you about. Now, let me introduce myself. My name is Thorsten Blatz, and I'm the Pharmaceutical Impurities Lead at a commercial department within RSSL. I work with a variety of departments to support our clients by solving their impurity problems. Daniel, can you introduce yourself?
Hello, everyone. My name is Daniel Nicolau. I'm a senior scientist here at RSSL, where I've focused for the last 8 years primarily in structural elucidations of unknown impurities in pharma products, working on extractables and leachables studies, but also the development and validation of LC-MS methods. I spent the last year focused entirely on our nitrosamine service, meeting the challenges that have been presented to me in the last year, some of which I will try to discuss with you today. So thank you all for joining.
So let me quickly talk about RSSL. So RSSL is a world-class CRO operating in the analytical space. The life science division of RSSL has recently refocused its strategy into a pillar structure, which will allow us to provide even better customer service. We are having a dedicated commercial leads looking after biopharmaceuticals, sterile manufacture, impurities, which is the area I'm leading, medical devices, which we hopefully introduce later in the year, and the core business, which really centers around the raw material testing and drug product release. Now, within my pharmaceutical impurities pillar, I'm focusing on the investigative pharma chromatography and microscopy department. My role is to work closely with the team to facilitate our clients' projects, offering a flexible approach and a mutual beneficial partnership. Now, throughout the year, I really like to run quite a few more impurity webinar sessions, which will introduce you to our newest instrumentations, such as the Bruker 600 MHz NMR, the 1290 Agilent Preparative HPLC, and the Vitek confocal Raman. And we also want to capture the LCMS and GCMS capability within the business. If after the webinar you would like to learn more about our offerings, please contact me direct via email or telephone, which can be found at the end of the presentation. Now let's get started with the webinar, and I hand over to Daniel to talk about nitrosamines.
I want to focus this webinar on a small, but I think important aspect of the debate and requirements surrounding nitrosamines in medical products, and that is concerning the factors within a risk assessment. I don't want to comment on which factors must be included in the risk assessment or how they would be applied in order to meet any regulatory requirements, but rather to highlight how these factors impact medical devices, medical products, and the method development and analysis of nitrosamines within them. We want to ensure that we generate the right analytical data to complement what is required within that risk assessment and in a timely manner. The first important question to probably ask within that risk assessment and the factors associated with it is determining the source of nitrosamines within a medical product. APIs have been discussed and published in great detail, with some having a greater propensity to generate nitrosamines. Ranitidine would come to mind and are probably very high in the list, but there are other APIs out there that may have that high probability, and will very likely be the main focus in a risk assessment. However, there are other sources of nitrosamines within a medical product. Excipients would be a prime example, or even the impurities within the excipients. There may be certain chemistry, functionality groups, and certain physical or chemical conditions that may allow the generation of nitrosamines to be included within a final medical product. So too with the solvents within the medical product or through the manufacturing process pose a potential risk within it. For example, using water may include sources of nitrogen, such as nitrites or nitrates, which may increase the potential risk of nitrosamines. So too would packaging potentially pose a risk. We know that some natural rubbers and other potential packaging materials include nitrosamines, and there may be a potential for those nitrosamines to migrate into a medicinal product. It may be that utilizing the guidelines within the regulatory frameworks surrounding extractables and leachables may be a suitable approach to understanding the risk packaging poses in regards to nitrosamines. And then it's the final product. Nitrosamines may form in your product over the lifespan of that particular product. And so it was likely worth considering that factor in understanding the risk. Again, I don't wish to determine what factors should be included, but rather to discuss how each of these nitrosamine sources impact the potential requirement of analytical testing and the challenges that are associated with it. It's important also to also understand how nitrosamines are formed in these various sources, API, solvents, etc. It is very well known that the reaction between a sodium nitrite and an amine under acidic conditions can form certain nitrosamines. So within the chemistry of your API, excipients, it's important to understand the presence of primary and secondary amines, and in conjunction with that, the presence of nitrate or nitrates in the formulations. And if any of those formulations with those particular chemistry, functionality, and species present falls under any acidic conditions at any moment in time, it may be worth considering how nitrosamines may be formed in your particular product. And then in conjunction with stability conditions, such as temperature, this too may play an important part. An example might be in a manufacturing process, the use of dimethyl formamide or DMF potentially decomposing and a further reaction with a nitrite to form the commonly found and known NDMA. As an analytical chemist, I'd say out of all those factors, the most significant impact on the analytical testing and the challenges it may potentially pose really focuses around understanding and calculating the suitable analytical thresholds. Within the regulatory guidelines for the FDA and EMA, there is a list of commonly targeted nitrosamines, and they have very well-defined acceptable daily intakes. So for example, on the slide on your left, as an example, you have NDMA, a very commonly targeted nitrosamine, which has a daily intake of 96 nanograms per day. You also have another highly targeted nitrosamine of NDEA, which has a smaller acceptable daily intake of 26.5 nanograms per day. And in order to associate that threshold in an analytical method, we need to calculate an analytical evaluation threshold. Some may use various terms, but ultimately I'm trying to convey what threshold we need to apply analytically on a specific medicinal product or API. And that can be calculated by taking that daily intake value for a specific nitrosamine and dividing it by the maximum daily dose of a particular API or product. So for example, in example 1, if you were attempting to determine the presence of NDMA in a product with an API that has a maximum daily dose of 10 milligrams per day, you can see that the analytical evaluation threshold is calculated at 9.6 nanograms per mg. So you can imagine, for example, a 10 milligram tablet, we would need to see 96 nanograms in that particular tablet. I can say from experience that that is a comfortable threshold, and it would allow me the flexibility to use a wide range of sample preparation techniques, as well as comfortably achieving that on any analytical equipment I deem fit for that particular example. But as you can see in example 2, things can get a little bit more challenging. If, say, for example, you're looking at NDEA, which has the lower daily acceptable intake value of 26.5 nanograms per day, in a product with an API that has a maximum daily dose of 4,800 milligrams per day, you can see that the AET drastically reduces down. And I can tell you that if that were to be 1 milligram in 1 mil, then that would be well below any LOD that can be achieved on any analytical instrument. Now, in this particular example, it's very unlikely you're going to have a low concentration of an API if the maximum daily dose is so great. So the likelihood is you're going to have a reasonably high concentration of API, which will be a challenge analytically to be able to extract out those nitrosamines. And in that example, it is also very likely you're going to have high concentrations of excipients. And having such high concentrations within a matrix will make it ever more challenging to extract out those nitrosamines from that particular product. And I can also say that the more nitrosamines you are trying to extract from a complex matrix such as that, the more challenging it becomes to incorporate that into one analytical method. So it's also important to understand that particular medicinal product in order to achieve the required analytical threshold that has been calculated for that particular product or API or excipient. And to do so in order to minimize the negative impact on the sensitivity of an instrument. Just because an analytical instrument can meet a particular LOD does not necessarily mean that is automatically achievable in a complex matrix. And so understanding the concentration of the API, excipients, which type of excipients, the pH of that particular formulation, so too will the physical state play an important role. Having a solid material from which to extract out the targeted nitrosamines might be a lot easier than working with a very complex matrix, such as a gel or a suspension. It's also important in that risk assessment to understand which nitrosamines you wish to target. It might be very easy for us to just automatically request a full screen of nitrosamines, but it's important to understand that if that is a genuine requirement, the analytical challenges will be far greater than if you are looking for just 1 or 2 specifically targeted nitrosamines. And then once you've understood which nitrosamines you wish to target, it's understanding the differences in the chemistry and the physical properties to your sample itself. So solubility differences will play an important role, and so too will pH. There are other factors that should be taken into consideration, but this is simply just an overview of the most important ones. And then once you've understood your product in relation to the API, the AET rather, the threshold that is required, you can start to utilize the various extraction techniques in order to prepare your sample for the analytical instrument. In the case of example 2 that I described, it would be very necessary in order to concentrate those nitrosamines from your sample in order to achieve the analytical threshold required. And so you would have to utilize a variety of techniques such as solid phase extraction, SPE, or even liquid-liquid extraction in order to concentrate those nitrosamines from your sample. I've just illustrated a very simplistic explanation of the SPE and liquid-liquid extraction. I know that liquid-liquid is actually quite a very old school type extraction, but it is incredibly useful under certain circumstances, especially in the sample preparation described within Nitrosamines in pharmaceutical products. And if you can utilize these particular extraction techniques with more commonly found techniques such as filtration, centrifugation, and precipitation, you can achieve that concentration of the nitrosamines while removing it and separating it from the complex matrix, the high concentrations of API and excipients. And then once you've understood how to prepare your sample adequately, the analytical chemist would have to decide which is the most appropriate analytical technique for a particular product. And they fall into two main categories as a mass spectrometry. Mass spectrometry is probably the most important because in both examples that I described earlier, the thresholds are still relatively low or, in some cases, incredibly low. And so it's important to use the most sensitive techniques available. And so mass spectrometry becomes a primary way to achieve those targets. And again, they could be separated into two major sections: liquid chromatography mass spectrometry, or LCMS, or gas chromatography mass spectrometry, GCMS. And within that, again, there's a wide variety of instruments that are available. And in both cases, the analytical chemist would have to decide which ion source is appropriate. So in the case of LCMS, you have atmospheric chemical ionization, or APCI, for usually used for smaller nitrosamines such as NDMA and NDEA. However, you can also use the more commonly found ion source of electrospray ionization, ESI, for the larger nitrosamines or product-specific nitrosamines, API-related, excipient-related. And again, in GCMS, a decision needs to be made on which ion source would be appropriate. Electron ionization, or EI, will generate a very unique pattern, fragmentation pattern, that is specific to a nitrosamine and can be compared to very well-known databases. However, if you wish to look for the molecular ion of a particular nitrosamine, a more softer ionization, chemical ionization, or CI, would become a far more appropriate ion source. Then again, once you've decided on that particular system, you would utilize two different types of mass spectrometry systems: triple quadrupoles, or TQs, or time of flight tops. Normally they come as quadrupole time of flight, or Q-tops. For the TQs, they are normally utilized for targeted nitrosamines at the lowest possible thresholds. These will be the systems that will be able to achieve the very low analytical thresholds that are required. However, if you are in the initial stage where you might wish to look for a wide variety of nitrosamines or a screen, you can use the high resolution mass spectrometry qualities of a time of flight to provide some high selectivity. So it's important when those factors are debated and described in the risk assessment that they will have an impact on which sample preparation, which analytical techniques would apply in those particular circumstances. So here's just a couple of examples and the challenges that we've faced and how we've developed some solutions in order to achieve that. What I would say is that the analytical method on the LCMS or the GCMS is a challenge, but relatively easily achieved. What I've found in the last year in developing these analytical methods for a variety of clients is the sample preparation and how best to complement that to the analytical technique and achieving the required analytical threshold for that particular product. So, for example, we've been able to achieve looking for NDMA in a solid API. And the challenge there is, how do I separate out, how do I remove the NDMA from that particular API? And as I described earlier, solubility between that nitrosamine and the API might be a very important factor to take into consideration and to take advantage of. So if the API, for example, is highly water soluble, you could use an organic solvent such as dichloromethane or DCM to dissolve that nitrosamine, NDMA, concentrate, and you're able to analyze a much cleaner sample ready for the analytical system. If, say, for example, that particular API is soluble in an organic solvent, rather, you could utilize water. In both cases, NDMA is soluble in both dichloromethane and water. Another example might be that you want to look for a range of nitrosamines in water that would be used for a range of medicinal products. And so the challenge there is trying to meet as low a limit of detection as possible. So we're trying to concentrate those nitrosamines in that particular water in order to provide or achieve the LODs required. And so a solution could be to use a solid phase extraction cartridge, an SP cartridge, to concentrate the nitrosamines on the solid phase, and then you can elute them, potentially with a different solvent, in a smaller volume, thereby concentrating those nitrosamines within the water. Another example would be trying to achieve the analysis of a range of nitrosamines in a formulation with a very high concentration of API. Now, I would say these examples aren't theoretical, they are very much real, and we have been able to achieve the development and validation of methods for all these particular examples. And the last example here has been seen to be, at least in my opinion, quite a complex method development. Because in this example, we've had very similar pKa and solubility values between the range of nitrosamines and the API. So it's been quite a challenge in order to find an appropriate SPE cartridge to bind the API while removing the nitrosamines from the product. That, unfortunately, has caused the nitrosamine to be highly diluted in the final solution, which we then had to prepare an additional sample preparation, separating out those nitrosamines in order for analysis. So it's important to understand that when those factors are described in the risk assessment, if you are looking for a range of nitrosamines in a complex product, the challenges and the time it would take and expertise required will be that much greater. So the take-home message is to understand that the factors included in that risk assessment, the source of the nitrosamines, the threshold calculations, the nature of the sample itself, and which nitrosamines you have decided within that risk assessment that require some analysis, how they impact the analytical challenges, such as the extraction of those nitrosamines, mitigating the impact of the matrix, while still achieving that limit of detection required in order to meet or provide and generate a validated analytical method. So thank you all for listening, and I think then we are in a position to take some questions.
So yeah, thank you, Daniel, for providing a better understanding about the risks in the nitrosamine analysis. So we have a number of questions coming in, and I'd like to ask a few in the time remaining. So what time of the development the analysis is required in phase 1 or only previous to registration?
I probably can't give you an exact determination. What I would say is the nature of nitrosamines and its toxicity is such that I would probably recommend analyzing or investigating the risk nitrosamines may potentially pose as early as possible in the development of a particular product. I think that's probably the most safe approach, I think, regarding nitrosamines, given the level of toxicity that they pose.
Okay. We've got another question from Annette around nitrosamines in biopharmaceutical products. Can you comment on the problem in this area?
Yes, I'm not fully experienced with biopharmaceutical products. However, the risk will still be present, but the factors may change. I certainly am not an expert in the risk assessment procedure for biopharmaceuticals compared to smaller molecule medicinal products, but the risks may still be present, and it still may require some analytical testing. the risks may be more focused on certain areas, and therefore it may be that your analytical testing, or the required analytical testing rather, would be more specific to that particular risk.
Chris has got another question around the recoveries when it comes to the sample prep between the SPE cartridge and the liquid-liquid extraction. Can you comment on the recoveries in this area?
Yes, of course. One of the challenges within that sample preparation is in order to achieve a suitable recovery from a complex matrix. And it does depend on the particular product. SPE, especially if you are binding the nitrosamine onto the solid phase rather than the other way around in trying to bind the API, as an example, by diluting, can generate relatively high recoveries. So too can liquid-liquid, especially if the solubility difference is strong. The difference is great. Then you will have a relatively high recovery value. Now, one of the best ways, I suppose, to be able to achieve high recoveries, irrespective of the matrix complexity or the technique chosen for sample preparation and analytical system, is to use internal standards in that method development. So using, for example, deuterated species would be a great way in order to mitigate not only a small decrease in recovery, but any variation in that recovery, so that any replicates of a particular sample, you would be able to achieve a consistent result, and you can demonstrate that the method you have developed and validated is more robust than, or as robust as you would require it to be.
Okay, thank you, Daniel. I don't think there are any more questions. Are there more? Oh, hang on. I don't think there are any more questions. So, hang on. Sorry. So why nitrosamines need to be separated from the API? Another question.
I suppose the main reason is that the analytical techniques that are normally utilized for these type of analysis, namely mass spectrometry, because of the nature of the sensitivity that they are able to achieve, they are even more sensitive to any potential interference from other species within a sample. And so high concentrations of API or excipients will play or have a potential negative impact. Even though we may be potentially using some chromatography techniques such as HPLC or separation by GC, there may be a potential that one of or two species may coelute and interfere with the ionization of a particular nitrosamine for the mass spectrometry. And that can have quite a significant impact on the sensitivity. And given that in some cases we're looking at very low concentrations of nitrosamines, even a small difference can have, or a small increase in concentration of an API, for example, as the question has been stated, can have a significant impact on that sensitivity. So it's important in that sample preparation and the development of the analytical method to take advantage of all the different separation techniques to analyze as clean a sample as possible.
There's another question around biologicals. Have you had much experience in the methods of biological products? Question from Kevin.
I'm terribly sorry, Kevin. I don't have an extensive experience in those. What I would probably say is that there are a number of sample preparation techniques within the biologicals that would aid in developing a suitable method for nitrosamines in that type of product. It'd probably be quite important to understand which nitrosamines in that product need to be analysed for, and so that would make have a important piece of information to determine how best to analyse or extract those nitrosamines from a biological product. I would probably say in that certain circumstance, the solubility or even the potential degradation of the product, proteins, antibodies, etc., would have allow other sample preparation techniques to be utilized in that case.
Okay, there's another question from Suresh. Do you have any defined % recovery of nitrosamines from the?
So I can give you some examples. So in some cases, I've been very fortunate that I've been able to find an appropriate physical difference, solubility, that I've been able to extract out the solvents and demonstrate 100% recovery. I wish I could say that that happens all the time, but it certainly doesn't. And in the example that I described in determining the threshold in one of the slides, in the API that had a very high maximum daily dose, it required multiple sample preparation systems to be put in place in order to achieve that. And the recovery was well below 100%. But because, as I've described previously, using internal standards, we were able to demonstrate that we were consistently able to extract out an appropriate amount of those nitrosamines to be detected at the threshold for that particular product. So it's not necessarily that you always are expected to get 100% recovery. Obviously, it would be highly desirable, but we have analytical systems in place to accommodate slight variations or decrease in recovery.
Now, another question from Juha. Do you have expertise regarding risk of nitrosamines in different blisters containing nitrocellulose and how to detect them, especially with headspace GC high-resolution mass spec?
Unfortunately, I can't give you an exact answer right now. We are in the process of trying to meet that particular challenge. It has been a little bit difficult to do, so unfortunately, I can't give you an exact answer. However, if you wish to send us an email, we would be happy to continue the conversation at a later date, and hopefully we'll be able to find a solution to your particular problem.
Okay, Peter has another question. What about the determination of nitrates in?
Was that the determination of nitrates in water?
Yes, it was. What about the determination of nitrates in water?
So for my experience and the literature that I have read, the presence of nitrates is, or nitrites, both, I think, do play an important part in aqueous solutions. I can't be certain, I wouldn't be comfortable to provide a certain concentration that could then be deemed as high risk or low risk, but the determination of those particular species will play an important part. It may be that actually you don't need to analyze for specific nitrosamines if your risk assessment highlights that the presence or potential presence of nitrites may be the root cause in any potential synthesis. And thereby, by looking at the nitrites, you may be able, within that risk assessment, to mitigate that potential risk, or at least describe it as a low risk.
Okay, there's a question from Ninesh. If we perform nitroamines once for all the RMs, do we need to perform on periodic basis, even if there's no change in product manufacturing?
Again, I can't give you a definitive answer. What I would say is that if you feel that the initial data did not fully mitigate or allow you to conclude that the risk was had been described and decided as negligible, it may be worth, especially in the stability of a particular product, or if you think there are any changes in manufacturing or the sources of batches of API or excipients, it may be worth having a routine analysis set in place in order to provide some data to describe that risk. Again, I don't want to explain or determine what factors have to go into that risk assessment or how to perform that risk assessment, but certainly if there is a present, even though there may not be necessarily any nitrosamines present in a particular product at a certain moment in time, that might not fully describe the risk described in a risk assessment.
Kevin has a question around validation. How much validation do you typically do for screening samples?
My experience so far with the screening of samples is an initial testing. A particular client may want to just have a very some analytical data to provide which nitrosamines might be present, even at low concentrations. And so using analytical techniques such as a QTOF LCMS will be appropriate. In regards to the validation itself, I would probably recommend, instead of validating a screening method, that we would conclude by targeting a range of nitrosamines and validating those targeted nitrosamines rather than a screen. It's quite difficult sometimes, especially if you're having wide variation in a particular product, to validate a particular sample and then screen in order to determine what nitrosamines might or might not be present, given that we have very well defined safety concern thresholds or daily intake values for those particular nitrosamines. I think the screening would be more of an investigative tool to then validate a more targeted method, would be my opinion.
Okay, another question. Tristan asks, does the risk assessment follow industry guidelines such as BPOG or USP?
I'm terribly sorry. I don't think I'm in a position to best answer that particular question. My understanding around the risk assessments is ensuring that the chemistry around a particular product and the origins or the potential origins of the nitrosamines within that product have been described. But as to how that risk assessment is performed, I'm terribly sorry. I'm not in a position to answer that question.
Okay, Pupenda asks, are the risks higher with liquid formulations or with solid in generally?
I would probably feel comfortable saying that solids would probably or likely pose less of a risk than liquid formulations. But again, it will depend on the chemistry. So having an acidic formulation will obviously pose a greater risk than having a basic or neutral formulation. And again, that is, in my opinion, less severe in solid medicinal products.
A question from Olivia. This doesn't particularly relate to method development, but are you aware if there's a requirement for excipients to be tested for nitrosamines, or is it just testing of APIs at the moment?
It's my understanding that with that risk assessment, it's important to understand the medicinal product as a whole rather than solely focused on the API. I know that a lot has been published and discussed about APIs, and that is very likely to be the primary focus of any risk assessment. But there has been some discussion surrounding excipients, particularly impurities, such as nitrocellulose, as an example, that might potentially pose a risk. It does not necessarily mean that excipients in all products pose a risk, but they may be unique to your particular product.
Another question from Jenny. If a risk assessment finds there is potential for nitrosamines, but does not specify which, and the contribution may only be a trace amount. How might you tackle that?
I suppose the first question I'd ask is, what do they mean by trace? Sometimes when you look at levels such as ppm or ppb, that is an achievable target by LCMS. And if that is the case, we could potentially begin any investigation by doing a screening analysis. If in a worst case scenario, that trace genuinely means at the daily intake values or very low analytical evaluation thresholds, it may then require within that assessment to screen for all the recommended nitrosamines that have been described in the FDA and EMA published documentation. Again, it would be quite specific. So I'd be happy to discuss that in further, but I probably need a little bit more information in order to give you a better defined answer.
We've got time for one more question. Achim asks, what was the lowest LOQ you could achieve for a drug product having a complex matrix so far?
I will fully admit, you maybe have caught me out on that, is that in example 2 in one of the slides, I understand that you will all receive the slides later on in the coming days. That particular example of a threshold requiring a 0.006 nanograms per mg was possibly the most challenging of analytical thresholds. But that doesn't necessarily mean you've done that analytically. We've been able to achieve that by concentrating the nitrosamines in a volume of that particular product and then determine that analytically. So that was probably the most challenging. What I would say is our analytical systems, which are probably as good as the market can provide, you can see in the low parts per billion, maybe even a little bit lower. But again, because you have a complex matrix, it genuinely requires us to concentrate that sample up to raise it sufficiently high to mitigate any matrix effect. So in that particular circumstance, that was quite a challenge. But again, it is achievable.
Okay, thank you very much, Daniel, for answering all these questions. Any questions we didn't cover during these Q&A sessions, there were quite a few more, we will forward these questions to Daniel, and as I mentioned at the start, we will circulate these along with the slides. Now, as I mentioned at the beginning, we're running a little promotion. So our little promotion is very much around the nitrosamine. So we'd like to offer the first 5 attendees who bring their product portfolio to RSL a 10% discount. So this hopefully is quite an attractive little promotion. Thank you for attending our webinar. I hope that you have found it very useful, and we appreciate all the questions you have asked. On behalf of everybody at RSL, I would like to thank Daniel for delivering such an informative webinar, and for you all out there for listening today. Please do not hesitate to contact me if you have any questions or you need our support with any projects. I wish you all a great day and hopefully speaking to you soon.